So very good morning, my dear students. So in the general microbiology class, today we are going to detailedly discuss about the microscopic techniques for examination of microorganisms. So last class we were discussing the detail regarding the different type of microscopes and how you can measure the microorganisms using some different types of microscopes. So to observe the microorganisms or examine the microorganisms very effectively under the microscopes, you have to use certain techniques, right? To increase the contrast or increase the resolution or clearly observe the internal content of the microorganisms. So these are the certain essential procedure you should follow before observing these microorganisms under the microscopes. This is, we call it as a microscopic techniques. These microscopic techniques, so starting from isolation of the microorganisms and the preparation of the specimens from the samples, how you are going to prepare the slide or specimens to observe under the microscopic, right, under the microscope. So we can prepare, first we can prepare the wet mount slide, right? So the wet mount slide is a very basic technique to observe the external structure of the microorganisms. So by using a water, right? So for that purpose, use a dropper. You can use a dropper for this preparation of this wet mount slides. You need a slide. Maybe you can use a cover slide, the cavity slide and cover slip and a dropper, right? Dropper. So first to take the clean slides, right? To clean slide and use a dropper to place a drop of water on the center of the clean slide, right? So you have taken these slides, then to put a drop of water over the clean slide by using the dropper, then use tweezer to lay the specimen on the drop of water, right? So this is a tweezer or the forceps or some of these very delicate the delicate needles you can use to take the specimen from the culture or from any samples. If you are going to observe the microorganisms inside the plant or inside the host, then you have to take a very thin cut. Right? You can take a very thin specimen. For that purpose, you can use a pit. So pit may be anything inside, right? Inside the certain plants, we call it as a pit. Or if it is not available, you can use that regiform or not fully ripened papaya, right? So anything, whatever that it is very sharply, right? Very sharply cut up. So you can use as a pit, right? So keep the specimen inside the pit and make it a small, very minute, very thin slide cuts, right? Cuts. So put the all these cuts, narrow cut, very thin shallow cuts into the drop of water and take that from the water by using these tweezers. Put on the whatever the slides you put a drop of water on, over that, just immerse it, right? You immerse it and generally touch the cover slip to the edge of the drop of water. So but why? So you have to use it once you have placed the sample, then cut the one edge, place the edge, one edge of the cover slide, the touching the water, whatever, how you the IT people, who are especially the template, right? The template over the mobile phone, how they are sticking, right? How they are sticking. First, they will clean it, then they will put some the liquids, then place it in the one side and and continuously press it to remove the air without trapping of the air bubbles, then fully cover your template over the mobile phone. Like that, same principle you should use to put the cover slides over the specimen and over the slides without trapping a single air bubble. Without trapping a single air bubble. So once you have placed the cover slides, then you can take it and you can observe under the microscope. So you would have studied that how to set up the microscope and how to observe the specimen under the microscope. This is a one kind of a technique. Right, so how to put a cover slides? This is get a clean slide and cover slip from your teacher. Place 1 drop of water in the middle of the slide. Don't use too much of water to run off. Small drip, 2 drop or 1 drop is enough. Place the edge of the cover slip on the side of the water. This slide cover slip side is right. Cover slip side. I think you will study those things in the practical, right? This cover slip should touch the water, edge of the water. Then finally to lower the slide towards here without trapping of any air. Right, so you don't need to use the stage clips when viewing the wet mount slides. The wet mount slides, you need not to put the clips, whatever the clips available on the microscope. Why? So because this slide is fully covered, then the specimen not get damaged. Right, specimen not get damaged. And another thing, another type under the specimen of the light microscope, right, we call it as a staining. We are using the another type of microscopic techniques, we call it as a staining. You can use the microscopic techniques is starting from pith cutting. So you can use the pith to cut the shallow cut. It's a one kind of technique. Second kind of technique is you can use wet mount slides with a drop of water with the cover slides. No, you are not doing any staining on that two of those stages. The third technique is a staining. So certain microscopic organisms, organs, or the structures present in that microorganism could not able to see very clearly under the microscope without properly stain. Okay? So therefore, there are certain chemicals and certain liquids you should use to stain different parts of the or different components of the microscopic organisms, right? This is we call it as a this is a procedure, right? How to stain the material before observing the microscopic organism. First, the wet as like a wet mounted slides, you can take a clean slides. Over the clean slide, you put a small amount of water, right? Then you can use a liquid. If you are going to use a liquid culture, you can use a liquid culture, right? That is called as a spread a thin film of material containing microscopic organism over the slide and allowed to air dry. You are not doing the cover slides here, right? You are not doing, or you are not covering the slides. Just allow to air dry 2 to 3 minutes. Then fixing. So fixing of this microscopic microorganism over the slides. How you can fix it? You can move your slides very slowly over the Bunsen burner, right? Over the Bunsen burner, 1 or few seconds, then it will get fixed, right? Process that kills the microorganism and attach them to the microscopic slide. Fixing preserves and minimizes the distortion of the cells. Why you are fixing is for the preservation and to minimize the distortion, so damage of these cells, right? So you can do for the 2 type of fixation. One is heat fixation. That's what I told you, to pass over the Bunsen flame for several times, then it will get fixed because with the evaporation of the water or the material, it will get fixed. At time you can use the chemicals to fix. So normally we are using as a methanol because it's a quick evaporating material. No, very quickly it will evaporate. The samples will get dry. So without damaging or distorting microorganisms. If you are using the ethanol, then the organism will get disturbed, right? And the organisms will get distorted. So therefore, you can use a methanol. Then staining. So third step is the staining. So there are different type of stains you can use in different type of microorganisms. After the fixing, or sometimes some microorganism specific components, before fixation you should stain. You will study in the coming slides, right? The staining means coloring microorganism with a dye that emphasizes certain structures. That's what I told you. For the clear observation of the certain structures of the microorganism, you should use a specific chemical to colorize this specific component or specific structure of the microorganisms. So before staining, the sample it must be fixed. That's what the procedure. First two procedures, smear, then fixing, then you are going for staining. The stains, specific stains, we are talking about stains, right? Stains are salts composed of a positive ion, cation, or the negative ion. Cation or anion. We call it as a cation or anion. Cation is we call it as a positive ion. Anion is also a negative ion. So why we are using that? Certain structure of the microorganism can accept positive ions. Certain microorganism structures cannot accept. That is we call it as a negative ion. If it is accepting, then you can classify the microorganism in different group. If it is not accepting, that we are classifying the microorganism in different group. Gram positive and gram negative. You would have studied in the theory classes, right? Or in the advanced level. The colored ion is called as a chromophore. The colored ion we call it as a chromophore. So there are two types of dyes commonly used in the microbiological studies. One is we call it as a basic dyes. Other one is we call it as an acid dyes. The first person who used these dyes and identified and classified the microorganism in bacteria into gram positive and gram negative, the person is we call it as a Gram. The scientist name is called as a Gram. That's what we call it as a Gram staining. Right? Gram staining. This can differentiate the bacteria in two groups, gram positive bacteria and gram negative bacteria. Is in the medical sciences who confirm the first. Right? Confirm the first. So the students, you are studying the central microbiology, don't worry about you should study any kind of microorganism. Sometimes you will get a job in the medical laboratories. Right? If you are specializing in general microbiology, right? Then you should know the old medical microorganism and agricultural microorganism, the whole other general microorganisms, right? So basic dyes coloring we call it as an chromophore is in positive ions. Chromophore, you all know that colored ion is called chromophore. If the coloring is we call it as a positive ions, chromophore is in positive ions, the basic dyes. Most commonly used dyes, right? So this is the dyes commonly we are using as a most commonly used dyes as a crystal violet, methylene blue, and saffron. So these are we called as in basic dyes. This basic dye is nearly pH range of 7. So that is we call as a neutral pH. So this bacteria are slightly negatively charged at pH 7. Bacteria will get charged negatively and pH 7. Therefore, this base by using this basic dyes, you can differentiate the gram positive and negative bacteria. Especially gram positive bacteria will not get stained by using this basic dyes. They need acid dyes, right? They need acid dyes. Therefore, the gram negative bacteria, if the bacteria get stained, we call it as a gram negative bacteria. Crystal violet, methylene blue, and saffron. Acid dyes, the color is a negative ions. Acid dyes are we call it as a negative ion. Stain the background, negative staining. Stain the background, not the microorganism. Microorganism will not get accept this dyes. So therefore we call it as a negative staining. Background will get stained. Organism will not get stained. Bacteria don't stain with the acid dyes and used to observe the cell shape, size, and capsules. So therefore, if you want to observe the what size of these microorganism, what is the shape of the microorganism, what is the capsule size and the consistency of the microorganism, or especially bacteria, you can use the acid dyes. These acid dyes are ecosin, nigrosin, and India ink. There are three types normally, three types of dyes commonly used by the scientists. We call it as an ecosin, nigrosin, and India dye. These three dyes, the bacteria will not get in. This is stay in the background. So therefore, the shape, the morphology, external morphology of these microorganisms, you can clearly observe by staining this microorganism by using this acid dyes. If you are using that basic dyes, definitely the internal content of the microorganism, because this organism will get this dyes. Then there are two types of staining method. One is we call it as a simple stain, other one we call it as a double stain, other one we call it as a complex stain. There are three different categories, right? Simple stain is an aqueous or alcohol solution of a single basic dye. Single basic dye, saffronin, crystal violet, and methylene blue. These are the basic dyes. So alcohol, if you are mixing this dyes in alcohol solution and stain the bacteria, this is we call it as a simple stain. Only one time you are staining. That's what we call it as a simple staining. The primary purpose is to stain entire microorganism to view the cell shape and basic structures. That's what I told you. This organism, you are stained with this basic dyes, the stain will penetrate inside. So the basic structure of the microorganism you can observe very clearly. The procedure is stain is appeared for a certain time, then washed off. Take that. Once you have fixed it, the slides, then add some amount of dye, keep it to 1 or 2, 3 minutes, then remove that dye by washing water. If the bacteria is accepting, stain will be present. If it is not accepting, stain will be washed off. So after washing off, you can put under the microscope, you can observe under the microscope, then clearly you can see what changes has been made by adding this dye. Right? So modern. So this is also, the modern days may be used to increase the stain intensity, increase the affinity of the stain for the specimen. Slide is dried and examined. Stain is put it certain time and wash off, stain dried and observe under the microscope. Example, saffronin, methyl blue, crystal violet, and carbol fuchsin also you can use as a basic dyes. There are 4 different type of basic dyes. Saffronin, methylene, and crystal violet commonly everywhere you can avail. The saffronin you can use for any dead material. Even plant samples also you can get using this stain. And carbol fuchsin also you can use as a basic dyes. So differential stain or double stain, which is the differential stains or double stains, is react differently to different types of bacteria. This staining, I told you, right? Certain bacteria positive, certain bacteria negative. If you are using this differential stain, certain bacteria will get accept, certain bacteria will not accept these dyes. So therefore, you can categorize. If you are diagnosing the unknown bacteria, you can easily categorize. Okay, this is negative, this is positive. Once it is categorized clearly in 2, then you can go for a deep examination. Oh, which bacteria may be from gram negative, or this is which bacteria from this gram positive. Right? So react differently to different types of bacteria can be used to distinguish among the different groups of bacteria. That's what I told you. Different groups of bacteria. Yeah? There are 2 different important differential stains used in microbiology. There are 2 differential stains. One is gram stain, other one is acid fast stain. Clear? One is a gram stain, other one is an acid fast stain. So gram stain, how to do gram stain? Nice. So developed by the Hans Gram. That's what I told you. The Gram is a scientist, is a Danish microbiologist, right, who discovered and developed a methodology for this gram staining procedure. The most useful staining procedure in medical microbiology, I told you, is first person who discovered this microorganism under the medical field, the gram, by using these different stains. That is your role. As a technologist, your role is to identify the new things, right? New things, technologist. That distinguishes bacteria of two large and medically important groups, gram-positive bacteria and gram-negative bacteria. From this invention, so you can use any kind of bacteria available in the earth. No need to differentiate medically important bacteria. Any kind of bacteria you can use, gram-positive or gram-negative. Provides useful information for disease treatment, right? This is useful for the disease treatment. So according to the classification, immediately if you want a solution, so you can sort it out, quickly sort it out which group of bacteria, so which type of medicine you have to take, or which type of treatment you have to give to plant, if the plant dies, right? Steps in gram stain. One is, there are two, I told you, there are two type of dyes you are going to use it. So one is we call it as a primary stain. The primary stains are the crystal violet. Primary stain. It's a basic dye, you are going to use it after the heat fix smear with a basic dye crystal violet. Then wash off this crystal violet. Then allow for the dry certain times. Then add another dye, we call it as crystal violet iodine complex forms. Methyl blue, you can use it, right? Methyl blue. After smear is rinsed with the water and iodine moderate solution is applied. You have to use a crystal violet iodine. This is a dye, modern dye. So modern dye you can use as a second step. So if you are observing that, if then again you wash off it, if this dye is accepted, it is indicating the gram positive. If it is, this dye is not accepted, it is a gram negative. Then decolorize it. Again, slide is washed with alcohol to remove the stain. Gram negative cells, but not from the gram positive cells. This dyes will be removed from the gram negative, not from the gram positive. Right? So once you will use a basic dye, then wash off the excess dye. You will add the crystal violet iodine as a modern. So then allow for certain time again to wash off with an alcohol. If it is removed, that second modern dye, no color, it is an indicating that gram negative bacteria. It is not removed, and it's an indication of gram positive bacteria. Right? Gram positive, gram negative cells will be decolorized. Gram positive cell will remain purple. Gram positive will remain the purple. Right? At the counter stain, another stain you will use as a counter stain. Alcohol is rinsed off. Saffron is applied again. First a crystal violet, then crystal violet iodine. Again you will add the saffron. Saffron is a common basic dye. So observe the dead cells. So which will stain cell that were decolorized. If the saffron is, if you are adding, if that dye removed cell is accepting the saffron, is indicate that the gram positive remain purple, gram negative will stain in pink color. If it is a pink color, it is because saffron is a pink. The negative, so if the negative will accept it, and positive will not accept because it is already colored by purple. Then the pink color is an indication gram negative. Positive, this is because of gram positive bacteria. Purple because of positive bacteria. Right? So it's a table, so principles. Right? Table and principle. Gram negative bacteria and gram positive bacteria. If you are adding the different dyes, what is happening? Right? What is happening in the cell? And alcohol, so cell wall is dehydrated, shrink the purpose, so powers permeable cell walls and membrane decreases. That's what you are adding alcohol. Saffron is a color absorb the dye in the gram. This is the principle and summarized here. Then how gram staining differentiate the gram positive and gram negative cells? That's what I told you. So the gram positive cells have very thick peptoglycan cell walls. You all know the bacteria cell membrane cell wall is there, that is we call it as a peptidoglycan cell, mycopeptide and peptidoglycan. There are two components are there, mycopeptide and peptidoglycan. The gram positive bacteria, the peptidoglycan compound is higher, more than 60%. And gram-negative bacteria this is mycopeptide, the murine. So therefore, the murine is not accepting the second time we are going to as an iron. Right? So iodine crystal violet you adding is not accepting. But peptidoglycan can accept. That's what it's a gram-positive bacteria. It's consisting, showing the pink color. So purple color is an indication of this. So the purple color is an indication of positive bacteria. Pink color is in gram-negative bacteria. So now the chemotherapy application and limitation of the gram stain is chemotherapy. So the gram-positive cells with the very thick peptidoglycan cell walls, they are susceptible to penicillin and cephalosporin. Certain antibacterial, so we call it as an this one we call it as an drugs. So whatever the penicillin, tetracycline, amoxicillin, everything you are taking antibiotic now. If the gram-positive bacteria is infected in your body, then this the penicillin and the cephalosporin like that of certain antibiotics will not work. Sorry, the antibiotic will work because these gram-positive cells are susceptible. Right? Gram-negative cells are very thin wall and the lipopolysaccharide layers are resistant to these antibiotics. Then if the gram-negative bacteria is there, then doctors will not give these antibiotics, then they will give another antibiotic, some flagyl and other things, right? Flagyl, gram-negative bacteria. If your tooth is removed, doctor will give some flagyl. There is a problem in some of these digestive tract, then they will give the flagyl. So because they are a gram-negative bacteria, the anaerobic bacteria. Right? So that's a chemotherapy purpose. The limitations are, what is the limitation? Not all the bacterial cells stain well with the gram stain. Not all. It is just you can differentiate. It's not exactly different. Not some bacteria will not. The gram stain only works in young bacteria cells within a 48 hours. Sorry, 18 to 24 hours. It will work. More than 48 hours, it won't work. Because the peptidoglycan is will go to old culture, then it won't work. Peptidoglycan compound will get matured, it will not, dyes will not work. So therefore, only young colonies can work. That's what doctors, they will take blood sample and culture it within 24 hours. They will stain it and they will let you. Otherwise, they will not give any drugs, doses, antidotes. Right? Then acid-fast fusion, the second category. This is a gram staining. Second category, acid-fast stain. The basic dyes you are using. Acid-fast stain also you can use it. The person, the Paul Ehrlich, is a scientist, right? Who discovered the mycobacterium, so causing the mycobacterium tuberculosis, right? Tuberculosis and leprosy, so causing organisms, so genus Mycobacterium, and pathogen of this Nocardia. So Nocardia is a genus Nocardia this pathogen belongs to genus Nocardia, right? Mycobacterium tuberculosis, leproscopy, these are causing certain diseases in humans, right? These bacteria have waxy cell wall. Instead of peptidoglycan or mycopeptide, they have the waxy cell wall, which makes them difficult to stain. So you cannot stain, but that's what they will use the acid-fast stain. Okay? The steps of acid-fast stain. These are the basic steps of the acid-fast stain. The primary stain, decolorization, counter stain. The same principle here also they are applying, but in different dyes. However, the heat big similar with the carbol feature. It's a sort of basic dye. It's a red basic dye, carbol feature. They are used crystal violet, the gram staining as a first dye. But here you are using the Haber fission, red basic, right? Gently heat. After adding, you are heating several minutes to increase the penetration and retention of the dye. Then allow to cool and rinse with the water. Excess dye you are washing. But there you are not heating. But here you are heating, adding carbon friction and you are heating. Then do a decolorization. There you are adding crystal violet alcohol, no? You are adding crystal violet alcohol. So, but here decolorization by using the acid alcohol. Non-acid fast cells will be decolorized. Acid fast cells remain red. The acid dyes accepted. So bacterial cells remain red. Otherwise, so the stains will be removed by acid alcohol. So counter stain, again you are using the counter stain as a methylene blue. Earlier you used as a saffron as a counter stain, gram stain. But here you are using the methyl blue. It's an applied which will stain the cells that are decolorized. So decolorized cells again accept that methylene blue. Non-acid fast stains are stained blue. Acid cells are remain red. It's red and blue now. So red and blue is indicating that certain bacteria is vacillase consisting certain bacteria is accepting this carboxylation acid, carboxylation dyes and remain red for the after the continuous process, the completion of these whole steps that indicate that these bacteria can be accepting the acid fast chain. Others are not accepting the acid fast chain. Then you can easily differentiate these groups. Special stains. Other than the stains, you can also certain special stains to stain certain structure of these bacterial cells and fungal cells. That is we call it as a special stains. These special stains are used to differentiate the certain structures present in the bacteria. For example, endospore. Endospore is a spore. This means individual bacterial cells is modified as a spore, right? So especially the gram-positive bacillus bacteria. So during the reproduction time, the cells, they are forming a spores, we call it as an endospores. So individual cells so become a spores, reproductive unit. From that, bacteria will come arise. To overcome these harsh and environmental conditions, so the food is limited, so they will produce endospores. If you want to identify the endospore producing bacteria, definitely you can use some special stains. Certain capsules and certain bacteria, they are having the flagella. To identify whether the flagella is in the polar region or in the side region, how many flagella, which component present in the flagella, shape of the flagella, size of the flagella. If you want to detect, then you have to stain that bacterial flagella by using certain special stains. For that purposes, you are using that special stains. So endospore stain, how you can stain the endospore. So endospore is an extremely resistant. That's what I told you. To escape from the harsh and environmental conditions, bacteria are producing, individual cells is modified and produce it as a converted as an endospore. It's a very hard structure. So some of the gram-positive, I told you, bacillus, to protect them from the harsh and environmental conditions, heat, drought, chemicals, and radiations. So an ordinary stain cannot penetrate. Or whatever the crystal violet, carbon friction, whatever you are using, it will penetrate. So mostly used as an Cheshire full tone. It's called as an endospore stain. It's a different chemical. We call it as an Cheshire full tone. Is a chemical used to stain the endospore. Steps for the endospore staining. So the primary stain, what you are going, the same. Primary stain you will use as a malachite grain. Instead of crystal violet or carbon friction, you are using a malachite green. It's applied on the heat fixed simian, and steamed for about 5 minutes. Then you are putting in the steam, right? The steaming the 5 minutes, hot steam. The malachite green will penetrate the endosphere. Malachite green can penetrate the endosphere. Then rinse with the water for 30 seconds. Washing. Remove the green dye from the rest of the cells except endosphere. Then counter stain. Counter stain you can use. Always counter stain is suffering. Except this acid fast stain. Acid fast stain you are using the methyl blue as a counter stain. But other many stains purposes, saffron is a counter stain. The rest of the cells. Appearance of the cells of the endosphere, pink cells with a green endosphere. So saffron is giving a pink color. The endosphere is a green color. If the pink cells with a green color, it's an indication of endosphere is present. Right? Endosphere is present. It's an advanced microbiological studies. You can use this. And capsule stain. different bacteria, they are producing different shapes of the capsule, and also the composition of these capsules also different with the different bacterial groups. So therefore, if you want to stain the capsules, then you can, because the gelatinous covers of the top of the cell wall, right? So we call it as a capsule. This is a deciding factor of the virulence of the disease. So therefore, how pathogenic, we call it as a pathogenicity. How these bacteria are virulent to decide, you should know exact capsule structure. So therefore, you are using the capsule stain. The capsules are difficult to stain because they repel most stains. So because the capsule and are water soluble and are easily disturbed with a harsh temperature treatment. So this capsule is a water soluble one. No, immediately it will get destroyed. Negative stain is used to obtain a dark background. That's what the indica nigrosin. So indica or nigrosin so using to obtain a dark background, under the dark background. That's what we told you for the observation of the external structure. You can use the nigrosin or India ink. India ink. Cells stained with a basic dye is in saffronin, but capsules will in the light halo around the stained cell and the dark background. A dark field microscope, you can use it. If you are using the dark field microscope with these dyes, definitely you can get a clear picture of these capsules. And flagella stain. So flagella also different type of bacteria having different number of bacteria, different shape of the bacteria, flagella, different size of the flagella, and the presence of the flagella one side or the other side or the polar. If you want to determine, you can use a flagella stain. Flagellas are appendages used for the locomotion that too thin to see easily with a light microscope. It is very difficult to see light microscope. Therefore, if you are staining it, you can observe it. Staining procedures are difficult, usually involve using a mordant and coating the flagella surface with a dye or metal. Definitely, you are using a silver staining. That is, we call it as a silver staining, but it is very difficult and very dangerous and very poisonous. You have to be very careful to use silver stain. So number and arrangement of the flagella can be used as a diagnostic aids to categorize the bacteria. And if you want to develop you as a bacteriologist, especially, you can practice all these stains. So this is the microscopic techniques used to observe the microorganisms very clearly under a microscope. So these are the microscopic techniques, very important as a microbiologist or a technologist, technomicrobiologist, right? So this is the end of the section. If you want to ask any questions, please you can ask.